Good morning, everyone, and welcome to day 3 of the MIT AI Conference, The Future of Health. This morning, our first session is with Dr. Chris Gibson, co-founder and CEO of Recursion Pharmaceuticals, and our moderator is Anne Carpenter, of the Institute Scientist at the Broad Institute of MIT and Harvard. And welcome to our fireside chat.
Hi, welcome everyone. I'm Anne Carpenter. My research group is based at the Broad Institute, and I am connected to the subject of today's chat because I serve on the scientific advisory board of Recursion. What's interesting about our background, though, is that I have to say a public thank you to Chris and to his company, because the company is in large part based on this idea that I was having very early on in my research group, where we were eager to get as much information as possible out of images and apply them to drug discovery. However, being in academia, I could only take things so far. And so we'd publish proof of principle papers and we'd develop methods and so on. But it's been really delightful for me to see this company develop over the past few years and really bring to fruition a lot of some of the things that I had really hoped would work out. So they've done a lot of the hard work to make that happen. And so I'm very grateful for that. So Chris, if you don't mind giving us just a bit of an introduction about yourself and tell us how you became interested in images as a data source.
Absolutely. Thanks, Anne. Yeah, so I was doing my PhD as part of an MD PhD program here at the University of Utah back in 2009 and working on a specific rare disease called cerebral cavernous malformation, which probably most folks haven't heard of, but affects about twice as many people in the US as cystic fibrosis. So it's really quite devastating. And in working on that disease, after a decade in the lab that I was in, they had figured out exactly how the disease worked. We had a target, we inhibited that target, and it turns out we actually made the disease worse in the models that we were studying. And it was in that failure that we recognized a couple of things, both how humbling biology is, which I think is a universal, but also that the images of human cells in which we'd broken the gene associated with this disease, that they looked really different. And so I sat down and started coding up this amazing piece of software. And then like the next day I quit and I started Googling and I found your work, Anne. And even though you're moderating, I think folks would love to hear a little bit from you about where the idea came from to use images to understand biology.
Yeah, well, so when I was a postdoctoral fellow at MIT in David Sabatini's lab, I was trained as a cell biologist and a microscopist. And so my research was to try to figure out what controls cell growth. And we were using Drosophila cells. They have really sort of ugly looking cells that are a little bit tough to analyze. And so I hit a wall, it sounds like, just as you did. And I found in the computer science literature some really great algorithms that looked like they would work really well for my project. And yet, as a biologist, kind of a point and click biologist, I had no idea how to take it from there. And so I dove in, started meeting with graduate students at MIT, some of whom started working on the project. And in the end, it produced, we wrote together this software Cell Profiler, which is now used by thousands around the world, specifically for this high throughput drug discovery. And so the idea of that, I just needed the software to measure one thing in my images, which was cell size at the time. But it's become very clear, of course, not just through my own lab's work, but a community of folks that are working in this space. It's become really clear that there's just a tremendous amount of information in images. And I want to just sort of pause and let the audience think about this for a moment. So what we're talking about is we take cells, we put them in a dish, we stain them with some stains, we take their picture, and somehow there's enough information there to tell us whether a certain disease pathway has been affected or whether a drug has been treated onto the cells. And that's pretty remarkable. I think at the beginning, when I was doing my project, it was like, great, we can measure the width of the cell and the height of the cell, and we can measure how much green stuff it has and how much blue stuff it has. But over time, as this field has matured, and especially now using deep learning, it's become really astonishing the power that this data type has. So I would love if you could just give our listeners a thumbnail sketch of what is Recursion's, what do you actually do? So you use images as a data source, somehow medicines come out the other side, but what's in the middle?
Yeah, so what we built at Recursion, initially based on all the great work that you did, Anne. In fact, we use Cell Profiler as part of Recursion until really, really recently for the main portion of our image analysis. But what we basically built is a giant factory full of robots where we conduct nearly 1 million different experiments every week in a variety of primary human cell types. And that's a pretty large scale of experimentation. We're able to actually do things like knock out every gene in the genome, explore massive libraries of hundreds of thousands or even one day soon millions of compounds. It's the same kind of scale that people do traditional high throughput drug discovery, but we're doing it using this image-based omics. And so at the end of every week, we stain the cells, regardless of the experiment, regardless of the biological question that we're asking, in this same basic way. And then we analyze them using computer vision and machine learning. In particular, we use a type of machine learning called representation learning. And we use that to create essentially a mathematical representation of biological state. And that allows us to ask lots of questions. And one of the primary questions we ask is, if some disease makes cells look different in many complex ways, is there some potential drug that makes those cells look healthy again? And in fact, we can ask this question for changes that no human can see. And we can ask it at the scale of 1 million experiments a week. And that's really exciting. And so the company's early days have been brute force search, taking hundreds of thousands of chemical compounds and adding them to diseased cells and looking for the ones that work and make the cells healthy again. And then we follow those up and take them into clinical trials. But over the longer term, we've actually now done nearly 33 million experiments, and all of those data actually relate. And so we're starting to try to fulfill our longer term vision, which is to build a map of cellular biology where we can understand how every gene relates to every gene and how any drug can move biology in any direction across that space, so that we can one day hopefully get a lot more medicines to patients more quickly.
Okay, so we should definitely talk about that, because that's a really exciting vision that this field has had for more than a decade now. So I really want to get to that point. But I do want to get a sense of the scale that you have. So you said 1 million experiments a week is kind of your typical routine these days. How many people does that represent? And how many people on the wet lab side versus the data science side?
Yeah, so Recursion's about 175 people right now. And we do a lot more than just the high throughput biology side. We also have folks who are designing our clinical trials, finance team, legal team, all of those things. So it's quite a big startup in the biotech space. That said, our main facility where we actually run these nearly 1 million experiments every week is typically run right now with the pandemic, run by shifts of 3 or 4 people working together. And I think that speaks to the power of automation. And it also speaks to our ethos around how important it is to use robots to do these experiments, to create that relatability, because humans are incredibly important for thinking about the results. But all of us have our different wrist flick when we pipette, and it's just not scalable, or you don't create relatable data if we're the ones making it.
Yeah, neither scalable nor particularly fun for the person doing that.
That is right.
We can both attest to that. So Recursion has 4 drugs in clinical trials, and you started in what year?
We started right at the beginning of 2014.
Yeah. So 6 years in, you've got 4 drugs in clinical trials. I don't know if there is, I'd be curious if any of our listeners know whether there's any similar kind of biotech or pharma startup that can claim such a rate. It's clear now that it was a really good choice to pick images as your data type. But I'm curious, at the beginning, what were some of the, venture capitalists or pharma companies' concerns about this overall approach? So what was the criticism that you faced when you were getting going in this space?
I mean, the most common criticism we faced was, if this is such a great idea, why hasn't someone done it? Which is, I think, a criticism everybody faces when they build something new that seems like it might be useful. A couple of things also came out. So people wondered why images, and I think the primary reason that images work so well, and to be clear, I think you could build Recursion over again with other types of omics data sets. But images are incredibly inexpensive to generate. They're about 2 to 4 orders of magnitude less expensive to generate than a transcriptomic, metabolomic, or proteomic profile of cells. And what that means is from a data per dollar perspective, we're 100 to 1,000 times more rich. And so if you're a venture capitalist thinking about putting $100 million down on a company, it can be really exciting to imagine that that company can do 100 or 1,000-fold more experiments on that same dime. The biggest question people had, though, was, can you really get all this information out of images? And thanks to a lot of your work and other folks' work, and frankly, if you talk to people, the idea that in biology structure suits function is not that foreign. The entire field of pathology, for example, where humans have been looking at cells under a microscope to determine what disease we have, is a great precedent for this. And on the side, you have all the amazing work from Silicon Valley and computer vision, machine learning, and China and other places where what we've realized is that of all things that deep learning does well, image analysis is like right at the top of that list. And so this just is a perfect marriage of those two things. And we ended up being pretty convincing both to VCs and several pharma partners as well.
Yeah, and I guess in 2014, we couldn't have predicted that deep learning would have been so powerful and transformative. I suppose that was right around when things were starting to heat up a bit. So, so that really did turn out to be great timing. But I can attest, from 5 to 10 years ago, as we were pursuing this in my lab, we knew that images were cheaper than other omic data. And the question was, were they going to be anywhere near as good? And so I have to say there were many sleepless nights where we've, invested a lot of time working on projects and just trying to get things working and just wondering, like, is this a total waste of time? Of course, there's some things you can see in images, but is it really going to be that powerful? So when we started seeing data coming out from our experiments showing not only that, yes, you can do useful things with image-based data, but also that it was in many cases on par or even more powerful than mRNA profiles, for example, high throughput mRNA profiles. I honestly was really shocked and certainly quite relieved given how much we had worked on trying to make it work. So it's just really quite a development here. So I guess now looking out to the bigger picture, and as other companies are thinking about what kinds of data they want to leverage in this space, even though the other omics data types are more expensive, is it sensible to expand out to those other data types, perhaps using them in combination with images?
Yeah, absolutely. And so that's something we've been working on. Discovering medicines is not like a single step. It's actually hundreds of steps. And what we found is that images are incredibly powerful as the foundation of that first step, which is doing the initial stage of drug discovery. And in particular, the type of work we do and you do, this so-called phenotypic screening, allows us to actually explore mechanisms in biology that maybe aren't understood. We don't come in with a hypothesis. We know that if we break like the CFTR gene, that's associated with cystic fibrosis. And if the cells change in thousands of ways, some of those ways may be represented in the literature and some of them may not. And it's one of the huge benefits of working with this phenotypic approach. That said, there are lots of things that images miss. And we also like to get sort of orthogonal validation of what we discover with images. And so we've actually started deploying other omics-based approaches upstream of, or downstream, of this kind of initial screening approach. And so when we find a drug that we think is really exciting, we can kind of check our work and make sure that it is also exciting in some of these other omics-based approaches. And we can also use them to do things like help predict toxicity and other sorts of elements that are really important when you're developing new medicines.
Yeah, that makes sense. And I think what I've seen from pharma, you alluded to this initially in the resistance to the idea that this could work, is for a lot of pharma companies, if you want to cure one disorder, if you want to cure one disease and you have a team of 100 people to work on it, then it makes sense to make the absolute most customized, really rich biological assay system, and then test drugs in that assay system. And then you've got all these very customized follow-up experiments and so on, but it's just incredibly expensive. And what you're pointing to is that this approach of let's just recreate the disease model in a very simple, very generic cell system. We're not going to catch everything, but for some low-hanging fruits, we're going to capture a fair bit of, we may capture some low-hanging fruit, and we can proceed with that at a rate that's just really a lot faster. What is the rough proportion of disorders that you've tried to model that you are able to capture with image-based phenotypes?
Yeah, so it depends on what you're using to perturb biology. In our early days, we were using RNAi, and in a cell type of relevance, on the order of 10 or 12% of genes that you perturbed with RNAi would create a statistically significant, when you're accounting for all of the noise of RNAi, a statistically significant sort of phenotype. We've now moved to CRISPR, and we're much closer to 40 to 50%, maybe higher. And there's a lot, we're very conservative when you're building a disease model. So there's probably more there, but we try to be very careful. If you use cytokines, soluble factors, and other sorts of biologic perturbants, the rate is extraordinarily high. So it turns out that nature has created things that do a really good job of breaking other elements of nature, and when you use those sorts of things like proteins, it's really incredible. And the nice thing too, beyond just this idea that we can go faster across different diseases, you can relate the data across those diseases. And so, unlike certainly people learn a lot in the more traditional approach where you build the perfect assay and you spend 5 years and you really understand the disease, but the translatability of what you learn is a little bit less clear as you go across the next 10 or 20 or 100 different disease models. Whereas when you do this simpler but very similar approach across thousands of disease models, or across the whole genome, or across every secreted factor or protein that biology, the human body has created, then you actually can relate all of that data and you can use it to identify really critical hypotheses that may otherwise be missed.
Yeah, I can imagine you've probably found connections between disorders that may have previously been seen. Yeah, we've certainly seen that in our own research on especially some psychiatric disorders, that common themes among them, and
or even disorders that are thought to be one often look very different. Maybe there's a couple of classes when you start diving in with this kind of approach.
Yeah, so I think in the way that that oncology, it used to be you have lung cancer, and you have lung cancer, here's the drug we're going to give you. Now we look at what mutation do you have, and so on, in a much more molecular way. It sounds like you can take that approach across different kinds of disorders. I want to, again, broaden out to kind of the big picture pharma space. For a large scale pharma company that's been around for, let's say, 100 years or 50 years, is it pointless for them to gather up all their existing data to mine? So over the past few years, of course, every company has realized data is the future and we need to gather up all our data. And so is that pointless, or is it more efficient or cheaper or better to make fit for purpose data in the way that your company is? So you have the benefit of starting from scratch. You don't have to try to resurrect any historical data.
Yeah, I mean, the answer is it depends, right? It depends on how that data was acquired. It depends on how relatable that data is over time. It also depends even on the computer systems. I've talked to leaders in large pharmaceutical companies who don't actually think they could get all of that data together. It wasn't created in a way with an expectation that that's something they would want in the future. And so we have the huge benefit of being able to start from scratch, knowing that we want every experiment that will ever be done at our company to be searchable and accessible to our data scientists and our biologists. And so we've just created that from the beginning. That said, I think, and you've seen some amazing papers in this space where some companies actually have put a lot of this data together, and it's created really, really compelling results. So in some contexts, I think it could be really important.
Yeah, I think my personal opinion is for a big pharma company, it's lovely to have as much data as possible available in kind of a browsable format. So if a researcher finds this drug for this disease, they can kind of dig in and see, have we ever tested this drug in a mouse model? Do we have any, somebody's photograph of a Western blot from 1960 or whatever. So I think that's useful to have that kind of thing browsable. But I think for data mining and for machine learning, the data pretty much has to be pretty high throughput to begin with so that it's consistent enough. And it may not be as consistent as what you're churning out at Recursion week to week in the past 6 years, but I think if you can access high throughput data, and we've seen some great publications of researchers digging up high throughput data and making good use of it in different ways. I want to ask one more question before we turn to some of the audience questions. I want to focus not just on the technology and the science that we've been focusing on, but I really want to bring out some questions about social responsibility. So for academics, it's annoying, but academics often see going to industry as selling out. On the flip side, those who are working in pure technology companies might see moving to a biotech company as like becoming more noble. You're curing human disease and so on. So I'm curious if you can talk about, as you've started Recursion, it's hard enough getting things going on the funding and the science sides, but can you talk about how you've worked on culture and thinking about your social responsibility?
Yeah, absolutely. That's been a really important part of building the company, and in fact, one of like the core areas of focus for myself and the team. So some of the things that we've done recently, we actually did a pretty large set of experiments on COVID-19. And we actually have a preprint out and released a pretty large data set, over 300,000 images that we generated in the deep learning embeddings from that work. And we basically gave that all to the public in the hopes that others could even find ways to use it. And we committed to not taking any sort of commercial benefit from that work. And so that's just a recent example. But as a company, we've released multiple data sets. There was a huge Kaggle competition with almost 1,000 teams that worked on a data set that we released of about 100,000 images. We've got a huge data set release coming, and that I know you know about. And so we'll continue to do that sort of stuff. But beyond that, we want to build a culture. We've brought tech and biology and chemistry all together. We've had to create an environment where different people with different backgrounds speak basically a new kind of language together. And that's instructional into the importance of diversity from a scientific perspective. But we believe much more beyond that we really have to grow diverse and inclusive culture. So we're doing things like we turned our prior headquarters into an incubator. I think in the next couple of weeks, we'll announce 3 startups that are going to be like our kickoff startups. And the focus of that incubator is actually on underrepresented founders to make sure that they can kind of get through the valley of death early on. And so we do all kinds of stuff like that. And I think it really energizes the team as well to help try and create a future type of biotech culture that is representative of the way we think as members of the team.
Fantastic. All right, so turning to some questions from the audience, I think I'll rephrase it as this. If your family member or yourself had a particular disorder, what would you do? In the sense of how do you research it online? Okay, step 1, Google everything you can find about it. But how can you think about repurposing and informatics approaches for these rare disorders where there's not entire companies are not working on them, kind of are on your own to figure things out?
Yeah, so if it were me, I would actually go into our database and ask whether we'd already modeled the disease and whether there were already inferences about what drugs might be useful. That's what I would do. And in fact, we get emails from patients and patient families all the time asking for us to essentially do that. And we work with a variety of patient foundations to try and advance medicines, even for diseases that have not many treatments. But you can go look. There's stories. If you Google Matt Might, there's a great story about Engelai 1, and I think not being a patient that I know of with any sort of rare disease, I think probably Matt and his wife's experience is one of the most instructive as to what they've done, how they've created a community and brought scientists in. So I'd direct people there because that's a much better expert source than me.
Yeah, and the question mentioned Feigenbaum for Castleman.
Perfect. Another great one, yeah.
Great example. A question about the locale. So you mentioned a lot of what you're doing about social responsibility, starting an incubator locally. There's a lot of things that are really peculiar about Recursion. One of them is that you're not in one of the hubs on a coast. And so can you comment on starting up a biotech in not a coast?
Yeah, so we're based in Salt Lake City, and I always say that's my wife's fault. She was doing her medical residency here, so I followed her here. Then we started the company without thinking too much about it. And then when you get to 25 or 30 people, it gets hard to move, but it's turned out to be really fantastic. We recruit a lot of people into Salt Lake. The cost of living is much lower. And I think this goes for not just Salt Lake, but Austin and many other kind of cities that aren't on the coast. And today we're seeing with the pandemic how it's possible to do remote work, how it's possible to work together with a distributed team. Almost all of our 170 people are here in Salt Lake, and many of them really enjoy the outdoors. And Utah is pretty incredible. It's 4 hours to like 5 major parks, Zions or Yellowstone are all basically there for the weekend. So we've enjoyed creating something a little bit outside of the typical spot. And I fly to those places to raise money, at least pre-pandemic, and then I bring it back to Salt Lake and we spend way less of it. And it helps us be a little bit stealth when we want to be.
Yeah. Well, it'll be interesting to see if this trend continues with some of the other companies starting up. Well, thank you so much for your time, Chris. It's been great to chat with you this morning, and we'll turn it back over to the moderators.
Likewise, Anne. Thanks for all your leadership in the space.
Yeah, thank you, Dr. Carpenter of the Broad Institute and Dr. Gibson of Recursion Pharmaceuticals. We appreciate your time and your fascinating work. We encourage everyone to join our Slack channel for further conversation with speakers and attendees throughout the event. And for our viewers, please stick around for the next session. That'll start in about 5 minutes. Thank you all.